Some animals have their skeleton on their outside, which is called an exoskeleton. And an example of organisms that have an exoskeleton are insects. And in this picture, you can see that the cicada is molting out of its skeleton so that it can grow larger. These white strands that are coming out of the exoskeleton are actually the insect's trachea. So when they molt, they actually have to pull out their tracheal linings and grow new tracheal linings. So they even have kind of like a skeleton in their breathing tubes on their body. So muscles function as our biological motors. They generate force and produce movement. Fibers are elongated cells that make up muscles. They use ATP to generate force and change length during a muscle contraction. Muscles can produce forces many times an animal's body weight, which is important because it allows animals to accelerate quickly. Within fibers, there are two contractile proteins, and these proteins enable muscles to shorten and produce force. The two contractile proteins that we will be talking a lot about today are actin, which are thin proteins, and myosin, which are thick proteins. Striated muscles appeared striped, so that's what striated means, striped, under a light microscope due to actin and myosin being arranged in a regular repeating pattern. Two types of muscles in our body have striated muscles. Skeletal muscles, which connect to the body's skeleton to move the animal's limbs and torso, and cardiac muscles, which contract to pump blood. So in these two pictures, A and B, you can see the striated appearance with the stripes because the actin and myosin are arranged in a regular repeating pattern. Smooth muscles appear uniform under a light microscope, or they appear smooth, and that is due to actin and myosin being organized in irregular patterns. And places in your body where you have smooth muscles are the walls of your arteries, which help to regulate blood flow, your respiratory system, which helps to control airflow, and your digestive and excretory systems, which help to transport food and waste through your body. So it's our first out of class clicker question. So the first question for today is, striated muscles include which of the following? A, skeletal muscles, B, cardiac muscles, C, smooth muscles, or D, skeletal muscles and cardiac muscles. So pause the video and answer the first question in the quiz below. Okay, so skeletal muscles. Let's look at these in more detail. Whole muscles are made up of parallel bundles of individual muscle fibers. So if we're thinking about going from very large to very small with muscles, we start with our overall muscle, and then we have a muscle bundle, which you see here. Each skeletal muscle contains several fascicles or bundles, each with numerous muscle fibers. So now we're going down, we have our bundles, and now we're going down to our muscle fibers. And muscle fibers are elongated cells that have many nuclei on them. So you can see here, here's the very long, elongated muscle fiber with many purple nuclei in one cell. Each muscle fiber encircles many myofibrils, which are rod-like contractile organelles. The microfibrils contain myofilaments, which are parallel arrays of the actin and myosin filaments that cause the muscles to contract. So if we go from biggest to smallest, we start with muscles, and then we have muscle bundles, next muscle fibers, which are made up of many microfibrils, and microfibrils are made up of microfilaments, which are the smallest. So we're going to talk about myofilaments because they're what allows our muscles to actually contract. So there's two different types of myofilaments. The first are thin filaments, which we can see in this red and greenish color picture. The myofilaments that are thin are composed of actin. So actin are our thin myofilaments. And the way that I remember that is actin, T-I-N, is very similar to thin. It's just missing an H. So what happens for the thin filament formation is that there's two actin filaments that are arranged in a double helix. Tropomyosin is that green and yellow looking linear protein that runs in the grooves formed by the actin helices. In comparison, thick filaments are made up of numerous myosin molecules that are arranged in parallel. So while our thin filaments, the actin is in a double helix form, in our thick myosin filaments, you can see here each of these blue lines is one myosin, and they're all arranged parallel to each other. Each myosin molecule, so if we look at one myosin molecule here, consists of two long polypeptide chains, which are coiled together, and they each end in a globular head. So that's what these blue circles are here. They're globular heads. Sarcomeres are the smallest subunit of muscle contraction. And so if we're looking in this picture here, we have what are called Z-discs. So a sarcomere is from one Z-disc to the next Z-disc. The shortening of thousands of sarcomeres along a fiber causes the shortening and the contraction of a muscle. Actin-thin filaments extend from both sides of the Z-discs towards the midline. So if we're looking at part A in this figure, we can see that the actin are these red filaments and the myosin are the thicker blue filaments. And if you look at our Z-discs, which are these zigzaggy purple lines on both sides, we can see that the actin is attached to the Z-disc and it extends towards the middle, but they do not connect in the middle. It's important to know that actin does not connect at the midline of the sarcomere. In the middle of the sarcomere, that is where you find the myosin thick filaments. So they start at the midline of the sarcomere and extend out. The thin filaments overlap with the myosin thick filaments in two regions within a sarcomere. So you could see in this picture we have one region of overlapping between the thin and thick filaments, and here's the second region of overlap between the thin and thick filaments. In regions where there is no overlap between the actin and the myosin filaments, titin links the myosin filaments to the Z-discs at the end of the sarcomeres. So here you can see the titin, and it's connected to the myosin thick filaments, and that just helps to anchor that myosin filament in place. So if we look at a cross section, so if we cut the sarcomere here and we put this under a microscope, we can see the thick and the thin filaments are arranged in a hexagonal lattice. So if we look here in this drawing of it, so here is our thick blue myosin filament, and there are 6 actin thin filaments surrounding it. So that's a hexagonal lattice. And you can even see it here in our cross section of an actual sarcomere. Each thick filament interacts with and slides with the 6 adjacent thin filaments. The precise geometry of the thin and thick filaments is critical to their interaction, and it's what allows our muscles to contract. So now we're going to watch a video just as a recap of how our muscles are organized.
Muscles are organized hierarchically, displaying multiple levels of organization. Let's start with the largest scale of organization, the muscle itself. Whole muscles in the body span joints. They attach to the skeleton at specific points to produce and control movement at a joint. The next level of organization is a muscle bundle. Whole muscles are made up of many muscle bundles. These bundles are surrounded by connective tissue and supplied by blood vessels. Muscle bundles, in turn, consist of many muscle fibers. An individual muscle fiber is a very long, slender cell with multiple nuclei. Individual muscle fibers are composed of long, rod-like structures called myofibrils. These myofibrils are arranged in parallel with each other along the muscle fiber's longitudinal axis. Individual myofibrils are made up of sarcomeres, which are arranged in series along the length of the myofibril. Sarcomeres are the functional units of muscles. Sarcomeres contain two kinds of protein filaments, thin actin filaments and thick myosin filaments. The actin and myosin filaments are arranged parallel to each other, and each set of filaments interdigitate within the sarcomere. Actin filaments extend from the ends of the sarcomere, called the Z-discs, toward the sarcomere's middle. Myosin filaments extend from the middle of the sarcomere toward its ends. Seen in cross-section under an electron microscope, 6 actin filaments surround each myosin thick filament. Muscles contract through interactions of myosin filaments with actin filaments. Skeletal and cardiac muscles have a striated appearance when viewed under a light microscope. Dark regions represent areas of overlap between myosin and actin, and light regions represent regions with no overlap.
Okay, so it's important to understand the structure of muscles so that we can understand how muscles actually contract. So sliding filament theory is what allows muscles to contract. So in this picture down here, we've just zoomed in on one sarcomere from one Z-disc to the next. And you can even see it here. We have one Z-disc and another, so those are those darker lines. Myofibrils contract to short lengths, and when you contract, there's the overlap between the actin and the myosin filaments increases, so more of them are overlapping. When the myofibrils stretch to longer lengths, the actin-myosin overlap decreases, so there's less overlapping. It's important to remember that the length of the myosin and actin filaments never varies. It's just the size in here, in that middle region, that changes. So the more overlap you have, the closer they are together, the smaller that gap in the midline is, the more relaxed your muscles are, the further apart and less overlap there are of the filaments. But the actual length of this red actin thin filament here is the same length as it is down here once the muscle contracts. So there's never any change in the length of the filament. It's just the space of overlap that changes. All the length change during muscle contraction results from the sliding of the actin filaments with respect to the myosin filaments within the sarcomeres. Because remember, these thick filaments are connected to the Z-discs through titin. So the thick filaments are not moving. It is actually the sliding of the thin actin filaments that is allowing for muscle contraction. The muscle's ability to generate force and change length is determined by the properties of its sarcomeres. Longer sarcomeres allow for a greater degree of shortening, produces larger but slower movements. Vertebrate muscles are shorter than some invertebrate muscles, so their length change is more limited, but it does allow vertebrates to move more quickly. So how does this contraction of our muscles actually work? So we now know that there's sliding of our thin actin filaments across our thick myosin filaments, but how does that actually happen? It happens through cross-bridge cycling. Along the myosin filament are two heads. The two heads of the myosin molecules bind to actin at specific sites. That binding, which you can see here, is what is called cross-bridging, because these globular heads of the myosin have attached to the actin thin filaments, so it forms a bridge or a cross bridge. The myosin filaments pull the actin filaments towards each other by means of these cross bridges. So having this cross bridge is what allows our muscles to contract. So interactions between the myosin and actin filaments is what's causing movement. So if we're starting here, we have this cross bridge that has occurred. The myosin head binds ATP, which lets it detach from the thin actin filament. So you see here our ATP has attached to the myosin globular head, forcing it to release from the actin. Then the ATP goes through hydrolysis, which it then forms ADP and 1-phosphate, and it causes the cocking of the myosin head back. The myosin head then binds to actin, forming that cross bridge. Once the cross bridge is formed, the ADP and phosphate are released, and it produces what is called a power stroke that causes the thin filament to slide relative to the thick filament. This sliding causes the sarcomere to shorten, which is what causes muscle contraction. So this is just another way to look at the cross-bridge cycling, just in case you like this picture better. I thought that they were both helpful. So muscle excitation contraction coupling is important when we're thinking about how cross-bridge cycling is working, because it's not just muscles that are going to contract. They need to have some input from our nervous system, which we learned about last lecture. So during relaxation, the myosin head cannot bind to actin. The attachment sites are blocked by tropomyosin. So if we look down here, we can see that at rest, tropomyosin is blocking our active sites. In order for the muscles to contract, troponin must move tropomyosin away from the binding site. So here we have troponin, it's going to move tropomyosin away from the binding site, and now you can see that these black circles on our actin, those binding sites are now exposed, which will allow the myosin globular heads to bind to the actin, causing that cross bridging, which is required for muscle contraction. This is regulated by calcium, which is released from the sarcoplasmic reticulum. The calcium will bind to the troponin, which will then cause tropomyosin to move away from the actin binding sites. To relax, the calcium is pumped back into the sarcoplasmic reticulum, and the muscle is at rest. So we're going to watch a video so that we can go over how this works and hopefully solidify it in your mind.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
So hopefully that video was helpful for you to visualize how muscles contract. In order for muscles to contract, we must have some stimulation from the nervous system. So for muscle contraction to occur, axons that extend from a neuron cell body branch to make synapses with muscle fibers. Some motor neurons that work with muscles will stimulate contraction by releasing acetylcholine, and that's what causes the excitation-contraction coupling. When action potentials traveling down a motor neuron arrive at the neuromuscular junction, which is the location, if we look at this picture, the location between where the neuron is and our muscle cells, the neurotransmitter acetylcholine is released into the synaptic cleft. So we learned last time that in order for this acetylcholine neurotransmitter to be released, it must be stimulated by calcium ions. These calcium ions then cause acetylcholine to be released, and this acetylcholine then stimulates the muscle to contract. So now it's time for some more out-of-class clicker questions. So the first question in this series is, ATP hydrolysis allows for what component of skeletal muscle contraction? A, the actin head to bind to tropomyosin. B, calcium levels in the cytoplasm to rise. C, the reorientation of tropomyosin and troponin. D, cocking of the myosin head to its high energy position, or E, detachment of the myosin head from the actin binding site. So I want you to pause the video now and go answer the second question below. All right, we have another question, yay! Which ion is responsible for the release of acetylcholine at the neuromuscular junction? Is it A, calcium, B, sodium, C, potassium, or D, chlorine? So again, pause this video and go answer the third question below. Awesome, one more question in this series. This question is, which ion is directly responsible for the initiation of muscle contraction? Is it A, calcium, B, sodium, C, potassium, or D, chlorine? Again, pause the video and answer the fourth question below. All right, so let's talk about muscle contraction from an action potential standpoint. The force exerted by a muscle depends on the frequency of stimulation by the motor nerve. As action potentials become more frequent, the amount of calcium that is released increases. This activates our cross-bridge formation in the muscle fibers. The more calcium that's released, the more active cross-bridge formation that occurs. A single action potential results in what is called twitch contraction of a certain force. The force depends on the type of muscle that this is happening in. And so if we see here, we have our muscle fiber action potential is reached from our motor neuron. There's a slight delay because it goes from the neuron and then it needs to then go into the muscle, and then we have our muscle contraction happening. If sufficient time is allowed for the muscle to pump calcium out so that the contraction ends and the force falls to zero, a second stimulus can elicit a twitch contraction of the same force. So anytime we're reaching this action potential, and then we have our muscle contraction, and then it falls back to zero, and our calcium is pumped out, then we can repeat that twitch contraction process. But just having a muscle twitch does not necessarily generate enough force for us to do something such as lift heavy weights. In order for that to happen, we must have force summation. And remember from last lecture, summation is just adding up of, in this example, the force. So if we just, like we learned in the last slide, we can have one twitch contraction and then it ends. But if a second action potential arrives before the muscle has fully relaxed, a greater force is produced. So this red line is showing just a twitch contraction, and then the calcium levels fall back to zero and the muscle relaxes. If we have multiple action potentials happening, coming from our motor neuron, these twitch contractions can sum together through summation, and it will create a greater force. When we reach a certain force, it's called a tetanus, and this is a sufficiently high stimulation frequency, and the muscle force reaches a plateau and remains steady as stimulation continues. So in this case, we have a sustained force. Measurements in animals indicate that skeletal muscles commonly produce tetanic contractions, which generate steady and large forces. Tetanus can also be induced by a bacterium, Clostridium tetani, so named because it can cause severe muscle spasms, particularly in the jaw, chest, back, and abdomen. Isn't that cool that a bacteria can do this? Motor units are the motor neuron and the population of muscle fibers that it innervates. So in this example down here in this picture, we have a green motor neuron and it is touching or innervating 2 muscle fibers. So that's one motor unit. The second motor unit in this picture is the blue neuron that is touching 3 muscle fibers. The size of the motor unit affects how finely a muscle's force can be controlled. So a motor unit can include relatively few to several hundred muscle fibers. So small motor units allow fine adjustments in muscle force. So a good example of a place in our body where we have small motor units are in our finger muscles, where we can do very fine scale things with our fingers, such as play a piano. Large motor units provide larger adjustments in force. So an example for humans is our leg muscles. We would not be able to use our legs to play the piano, but we can use our legs to do something such as kick a soccer ball a very far distance, where we would not be able to do that with our fingers. Together with stimulation frequency, a muscle's force output depends on the number of motor units, and therefore the number of muscle fibers that are activated. Within each motor unit, muscle fibers generally share similar contractile and metabolic processes, but differences in these properties can give muscles very different capabilities, such as comparing our finger muscles to our leg muscles. So we have two different types of skeletal muscles. We have slow twitch fibers and we have fast twitch fibers. Slow twitch fibers, which are shown in the red color in this picture below, are found in muscles that contract slowly, and they consume much less ATP to produce force. So slow twitch fibers obtain energy through oxidative phosphorylation, which occurs during aerobic respiration. Animals use slow twitch fibers to control their posture or to move slowly and economically. So animals that move slower have more slow twitch fibers or red fibers. Fast twitch fibers, also called white fibers, which are in this yellow color in this picture, generate force quickly, producing rapid movements. But because they generate force so quickly, they consume more ATP, which makes sense because if we're thinking about generating force more quickly, we must contract more quickly, which means that we have to have that cross bridge power stroke happening more and more quickly. And each time that happens, the myosin globular head will consume 1 molecule of ATP. In order for our fast twitch fibers to obtain enough ATP, they mainly get their energy through glycolysis, which happens during lactic acid fermentation. An animal, such as a predator, depends on fast twitch fibers to sprint or to lunge at its prey. Most skeletal muscles are composed of mixed populations of red or slow twitch fibers and white or fast twitch fibers. And this provides a broad range of contractile function, which helps an animal to survive. So let's talk about each of these types of fibers independently. So slow twitch muscle fibers develop force more slowly. But because they develop force more slowly, They have greater resistance to fatigue in response to repetitive stimulation, which means that slow twitch muscle fibers have a greater endurance. In order to do this, slow twitch fibers express a chemically slow form of myosin. So it has relatively low rates of ATP hydrolysis, but that limits the speed of cross-bridge cycling and force development. Fast twitch muscle fibers, in comparison, have fewer mitochondria and fewer capillaries, but they fatigue very quickly. This is because fast twitch muscle fibers express a chemically fast form of myosin, which has a high rate of ATP hydrolysis. This favors rapid force development and movement. White fibers are larger than red fibers, so white fibers generate more force. So let's think about how these different fibers might affect speed, and specifically looking at fish in this example. Most fish have a narrow band of slow twitch red muscle fibers that run beneath their skin, which they use for slow, steady swimming. So if we look at the mackerel here is our red slow twitch fibers, and you can see it's just a very small strip that's running along the side of the fish. Due to the evolutionary arms race between predator and prey, there has resulted in selection for specific muscle fiber types depending on the particular organism, and other musculoskeletal specializations that allow for rapid movement and maneuvering. So if we look at our mackerel, when escaping from a predator or attacking prey, so this is for both if you're a predator or prey, you're going to recruit your larger, fast twitch white muscle trunk area to help you have rapid swimming. Tuna and some sharks, which are predators, have evolved a deeper region of red muscles that actually is inside of their slow twitch muscles, which is different than what the mackerel and most other fish have with their slow twitch muscles on the outside. By having their slow twitch muscles on the inside, that enables them to keep their muscles warm and contract longer and faster, which allows them to move faster for rapid swimming, which helps them zoom through the water to catch their prey. So let's talk about skeletons now that we know all about muscles. You cannot have muscles work without some form of skeleton. So hydrostatic skeletons are found in nearly all multicellular animals. In hydrostatic skeletons, there's fluid contained within a body cavity. This fluid is the supportive component. It helps to keep the shape of that skeleton. Muscles exert pressure against the fluid to produce movement. So if we're looking at this sea anemone down here, here's our fluid-filled cavity, and we have our circular muscles here that reduce the diameter of the body cavity when they contract. And then we have longitudinal muscles, which help to contract. When they contract, they reduce the length of the organism. So let's look at earthworms for an example that is probably easier to understand because you've hopefully all seen an earthworm at least once in your life. Earthworms burrow through the soil by moving a series of hydrostatic segments along their body. So that's why when you look at an earthworm, it has this segmented appearance because each of those segments is a hydrostatic segment. So that's how it has its skeleton. So between each of these discs, we have a fluid-filled cavity. What happens when an earthworm moves is it will contract its circular muscle, which will make it shorter, and that force generates movement forward. Humans and other vertebrates also have hydrostatic skeletons, we have hydrostatic skeletons, for example, in our spine. So our vertebrae are an example of bones that have hydrostatic properties because there's fluid in the inside of them. Some animals have exoskeletons, which are rigid external skeletons. So exo means outside. So their skeleton is on the outside of their body. Exoskeletons have some advantages. They provide hard external support and protection for the organism, and it allows muscles to attach from the inside of the body. One other advantage for an exoskeleton is it helps to reduce water loss from the organism because they have that hard covering. Some examples of animals that have exoskeletons are some mollusks. So we have clams here, we have a nautilus, and arthropods. So that's insects, lobsters, crabs, and other crustaceans. The biggest disadvantage to exoskeletons is that it limits growth. So you can only grow as big as your exoskeleton if it's on the outside. In order to combat this, insects and other arthropods have to molt their exoskeleton. So here we see a dragonfly. It has molted out of this exoskeleton, and then it has a period of time where its new exoskeleton has to harden. During that time, it allows the insect to grow larger. So molting helps the insect and other arthropods get bigger. Humans and other vertebrates have endoskeletons, and endoskeletons lie internal, so inside most of the animal's soft tissue. There's major advantages to having an endoskeleton. The first being that vertebrates can grow extensively. They're not limited by the size of their skeleton that's surrounding their soft tissue. Instead, their skeleton can grow and allow for bigger and better growth. Another advantage is that when it's broken, the endoskeleton can be repaired. This is more challenging for organisms that have exoskeletons. There's two parts to endoskeletons. The first is the axial skeleton, which is in that yellowish color for both the human and the cat. The axial skeleton is the skull and the jaws of the head, the vertebrae of the spinal column, and the ribs. The axial skeleton provides protection for internal organs such as the brain, lungs, and heart. The appendicular skeleton are in blue on the human and the cat, and these are the bones of the limbs, which include the shoulder and the pelvis. And the appendicular skeleton provides increased ability for movement. So how are vertebrate bones formed? There's two different ways that vertebrate bones can be formed. The first is through direct formation. And in direct formation, precursor cells initially differentiate into osteoblasts. And these osteoblasts immediately begin producing bone. And this occurs in our skulls and our ribs, which are important places for this to happen because we need these bones to protect important organs. The next way that we produce bones is through endochronal ossification, which is an indirect formation. In this indirect formation, we first form cartilage. So if we look at step 1 here, initially the future bone is formed of cartilage. Then the bone starts to form where blood vessels penetrate, causing the cartilage to degenerate locally. So you can see here that we have these blood vessels, which is causing the cartilage to degenerate or break apart. As cartilage is further transformed into bone, new cartilage continues to be added at the growth plate, causing the bone to grow in length. So we can see here that here's our bone where our cartilage has degenerated, but we still have this area of growth that allows bones to continue to grow to a certain length. Due to this process of using cartilage to form bone, we then have different types of bone. The first is compact bone. Compact bone consists mainly of dense, mineralized bone tissue containing osteoblasts and the network of blood vessels supplying the osteoblasts with oxygen and nutrients. The compact bone forms the walls of the bone shaft. Spongy bone consists of small plates and rods known as tuberculae, with spaces between them. And that's why it's called spongy, because there's spaces, or if you look up here, it looks almost like a sponge. The spongy bone is found in the end of the limb bones and within vertebrae. Spongy bone is very important because it reduces the weight of the bone in these areas, which are typically where things such as joints form. But they are stiff so that forces are transmitted effectively from the joint to the bone shaft. Bone marrow is a fatty tissue found between the tuberculae and within the bone's central cavities. And bone marrow contains many important cells that is necessary for our bones to function properly. So I said that spongy bone is important because it's typically in locations where we have our joints. And there's two different types of joints in most vertebrates. The first are the ball and socket joints, which are highlighted in purple on this human skeleton. Ball and socket joints allow rotation in 3 axes, which allow for the widest range of motion. An example in our bodies that have ball and socket joints are the shoulders and the hip joints. The shoulder is the most mobile joint in the human body, which makes sense if you think about which part of your body you're able to move the most. Hinge joints allow one axis of rotation. An example of a hinge joint in the human is our elbows and our knees. Another example in other animals are ankles in dogs. Joints with a broader range of motion are less stable. So for example, our shoulders, which are our most mobile joint in the body, they have the most movement, are also the most often dislocated or injured. The ankle joints of dogs, horses, and other animals, for example, is a very stable hinge joint. And it's very unlikely that it will be dislocated, but its range of motion is much more limited when we're thinking about being able to flex and extend that part of the body. So how does our skeleton and our muscle systems work together to actually cause movement? Muscles can generate force only by pulling. They cannot push on the skeleton. And that makes sense if you think about how our muscles contract. They contract when we have our contraction of our sarcomeres, and there's nothing that has any pushing effect. We either have contraction or it relaxes and goes back to its normal position. So only by pulling or contracting can muscles generate force on the skeleton. Antagonist muscles are muscles that are arranged in pairs to pull in opposite directions. And this pairing allows our muscles to move our skeleton in 2 directions. We have flexion, which is the joint motion in which bone segments rotate closer together, and then extension, which is joint motion when they move apart. An example of this are our triceps and biceps. The biceps shorten or contract to flex our elbow joint, so we're going in an upward direction towards our shoulder. Our tricep is the opposite, and it shortens to extend the elbow joints. So here we have the bicep, it pulls our elbow joint closer to our shoulder, and when we contract our tricep, we're actually pulling the elbow joint away from our shoulder. And in this way, we are able to move our elbows. It is only because these antagonist muscles work together, pulling in different directions. This increases strength and it improves control of joint motion. So, our last few clicker questions for today. Why are muscles always associated with some type of skeleton? A, the skeleton provides a storage area for calcium ions used by muscles. B, the skeleton transmits the force generated by the muscles. C, the skeleton is always necessary to support the body against gravity. Or D, the skeleton is necessary for animals to grow. So I want you to pause this video and answer the 5th question below. Okay, one more question for today. Exoskeletons made of chitin protects internal organs and limits water loss, but has limited ability to grow and repair. Exoskeletons are found in which of the following group of animals? A, mammals, B, sponges, C, invertebrates, or D, vertebrates. Again, pause the video and answer the last question below. All right, I hope that you enjoyed that lecture learning about how animals move. And if you have any questions, post them on the discussion board, email me, or attend the Zoom office hours Tuesdays and Thursdays from 1 to 3 p.m. And I hope you have a great day.